Good afternoon, good evening, or good morning. It depends where you are to this third session about therapies for MPS. MPSs. I'm Hernana Martino from Buenos Aires, and I'm glad to be here chairing this very interesting session about the recent advances of therapy for MPSs. Next speaker, it's Dr. Stuart Ellison. Dr. Stuart Ellison is a research fellow in the Stem Cell and Neurotherapies group in University of Manchester. He is a PhD. He received his PhD in the University of Birmingham. Then he moved to the Imperial College in London, studying with the pioneer work with the lentiviral vectors for ALS and Huntington disease. Last year, he is joining the Dr. Professor Bigger's team in Manchester, and he has developed there several lentivirals mediated ex vivo gene therapies, and that's the subject of his lecture, ex vivo gene therapy for MPS IIIA, IIIB, and MPS II. Stuart, please, it's your time. Thank you.
Thank you. So I'd like to start by thanking the organisers for the opportunity to present today, tell you all about the work we've been doing developing stem cell gene therapy treatments for MPS IIIA, IIIB and MPS II over in Manchester. So I've been within the group for the past 9 years now, so I'd like to go through and discuss some of the data we've generated in that time. First, I'd like to start by giving a very brief background about MPS. I'm sure we're all aware that it's caused by a lack of lysosomal enzymes involved in the breakdown of glycosaminoglycans. MPS can be neurodegenerative or multisystemic in nature, depending on the type of storage material that accumulates. Heparin sulfate storage largely affects the brain, as seen in the MPS III subtypes, whereas dermatan sulfate and keratin sulfate accumulation largely affects the somatic organs. You can also see accumulation of both heparin sulfate and dermatan sulfate together, and this typically affects both the brain and the somatic organs, as seen in MPS I, MPS II, and MPS VII. The focus of our lab over the past few years has been to develop new therapies for MPS subtypes with a neurological component. So we've been focusing on MPS IIIA, B, and MPS II Hunter, and these all have very similar phenotype in that they affect children very early on in life, with progressive cognitive and later motor decline, often with severe behavioural problems, sleep disturbances, hyperactivity, and sadly there's no effective treatments currently available for these diseases. And these children don't typically reach adulthood. So the approach we've been developing is hemolytic stem cell gene therapy. Essentially, it's based on isolating patients' own stem cells and transducing them with a lentiviral vector to incorporate correct copies of the defective gene. Following a full conditioning regime, these genetically modified cells are transplanted back to the patients, where they can start to repopulate the blood system and start to manufacture the missing enzyme, cross-correcting cells in the periphery. Crucially, some of these cells can traffic up to the brain as monocytes, start to produce enzyme in the brain, where they can then begin to cross-correct affected neuronal cells. And since this is an autologous treatment, where you have genetic modification of patients' own cells, you don't have the risks associated with graft versus host disease, as seen with a standard allogeneic bone marrow transplant. And with this technology, we can have the opportunity to overexpress functional enzyme in the cells that traffic to the brain, thus increasing the chances of therapeutic benefits. And as we've heard from many other talks in this conference, and from Alessandra Beefy just prior, this technique has been used to great effect for MLD and also several other immunodeficiency disorders such as Wiskott-Aldrich syndrome and ADA SCID. So over the past decade, we've been developing stem cell gene therapy for MPS IIIA. We first published our proof of concept study back in 2013 in Molecular Therapy, where we were able to effectively demonstrate neurological correction in MPS IIIA mice following stem cell gene therapy treatments. We've since gone on to do a whole host of preclinical safety and efficacy studies as required by the regulators in order for us to progress into first-in-man clinical studies. And I'm pleased to say we started a phase 1/2 clinical trial in January of last year in MPS IIIA patients. So the focus of my talk today, I'd like to take you on that journey from the initial proof of concept studies, showing you some of the data from those animal studies, through to the preclinical data and some of the challenges we faced in order to translate this therapy to the clinic. And then I'd like to then show you some of the data from our first treatment patient that's been treated with this therapy and show you some of the data 2 years on. So first, for the proof of concept study, we have a lentiviral vector with a codon-optimized SGSH transgene. We compared a couple of different promoters, the human ubiquitous PGK promoter, as used in several other studies, and also a myeloid-specific human CD11B promoter. And the reasoning behind this is that we wanted to try and improve targeted transgene expression in the cells that traffic to the brain. For our experimental design, we isolated hemopoietic stem cells from MPS IIIA donor mice, transduced them with either of our lentiviral vectors, and then transplanted them back into busulfan-conditioned MPS IIIA recipient mice. And then 6 months post-transplant, we looked at a plethora of behavioural and immunohistochemical outcome measures. What we observed was that monocyte trafficking to the brain was effective regardless of promoter used. We typically saw 22% overall macrophage engraftment after 6 months following treatment. In the periphery, we saw SGSH enzyme levels approximately 5-fold wild type for both our treatment conditions. However, when we looked at the brain, we saw that our CD11B promoter was more effectively increasing brain SGSH levels of 11% versus only 7% with PGK. And what that looks like when we look at behavior in these mice, we did an open field 1-hour behavioral study. We can see that MPS IIIA mice, like the patients, are hyperactive and move around a lot more in this open field box compared to wild type mice. With 7% enzyme activity with a PGK promoter, we're only able to, the mice were similar to untreated MPS IIIA mice. So there wasn't much of a behavioral benefit in that treatment group, whereas our CD11B vector were able to correct to wild type levels in all the parameters we looked at in this particular model. When we looked at the brain in more detail, looking at immunohistochemistry in various areas of the brain, this is the cortex. We can see in neuronal cells, the swollen lysosomes characteristic of the disease, seen here in the with the green LAMP2 staining. These very swollen cells are greatly reduced with both our vectors, but more effectively so with CD11B. And when we look at the brain heparin sulfate storage in these mice, we've effectively normalized heparin sulfate storage with our CD11B vector, where there was only a partial correction with the PGK. Looking at the neuroinflammation, so we looked at activated microglia in the brain. Again, these activated cells are greatly reduced with our CD11B vector, which is more effective than the PGK. So, having shown successful proof of concept, we then discussed with the regulators and set up various experiments to meet regulatory requirements in order to translate this to the clinic. Most of the work revolved around evaluating the safety and efficacy of the lentiviral vector in human stem cells. So we've transduced stem cells at small scale and larger scale at various different vector doses. And we've also performed various biodistribution studies in humanized mouse models, which I'll talk about a little later. So this next slide shows a schematic of the process of a clinical scale manufacturing. So we had to develop a clinical protocol in the lab in preparation for future clinical trials. And essentially what we've been doing here is isolating stem cells from a donor leukapheresis unit on a Clinimax system, counting, assessing the cell viability by flow cytometry, and then pre-stimulating these purified cells overnight in a growth media with various different cytokines to allow more effective transduction the following day. We transduce for 24 hours before washing and harvesting the cells, at which point we cryopreserve the transduced cell product, which is effectively our investigative medicinal product. And we do a whole host of QC tests, we make sure the cells are pure and viable following transduction with flow cytometry. And we also seed the transduced cells in 14-day cultures to evaluate normal lineage development. And from this, we can do various different tests to check for transduction efficiency and overall VCN in the transduced cells. So one of the regulatory requirements was to show effective scale-up transduction with our GMP SGSH vector. So we had help from collaborators over in Boston, TransLab, who did this work. And we were transducing cells with a couple of different doses at the time, a single dose with an MOI of 100 and also a double dose of an MOI of 100. And these are really quite large doses, which were likely to be our maximum effective dose at that time. And with this study, we evaluated the vector copy number and enzyme activity pre and post cryopreservation as well, and showed no difference between the two. We then took this transduced cell product forward and used it to transplant into a humanized mouse model And the aims for this particular study were to demonstrate that these transduced cells successfully engraft, and then evaluate where the IMP distributes throughout the body, demonstrate efficacy. We also wanted to evaluate if there's any toxicity in the organs and confirm no vector shedding. When we looked at engraftment and biodistribution in these mice, to evaluate engraftment, we used an antibody specific for human leukocytes with no cross-reactivity to mouse leukocytes. And the highest engraftment was seen in the thymus, with no difference between mock and the transduced group. I should mention that this is a double transduced cell product. We saw lower levels of engraftment in the bone marrow, spleen, and the white blood cells. When we evaluated biodistribution, we looked at a wide range of different organs, and we saw a very similar VCN in these organs compared to the product that was transplanted into the mice. Importantly, we didn't see any of our transduced cells in the gonads or in some of the muscle cells, the gastrocnemius muscle. And then with this study, we've also demonstrated efficacy by looking at bone marrow SGSH activity. So yeah, this work completed a batch of preclinical data, but at this stage, transduction with a double transduction with an MOI of 100 was using very large volumes of vector, and we set about with our collaborators at Great Ormond Street Hospital, Adrian Thrasher, Karen Buckland and the team, who did a load of work looking at transduction enhancers to try and reduce the amount of vector required, so to make vector batches go a lot further for these studies. I haven't got that data to show you today, but I've got some data that we've been working on with our MPS 2 GMP vector products. And for this particular study, we've done small-scale transductions at a range of different vector doses with and without transduction enhancers. So for this, we've used lentiboost and protamine sulfate and a cryopreserved CD34 stem cell products isolated from mobilized peripheral blood. And what we can see at the end of the 14-day cultures, when we evaluate transduction efficiency by qPCR in the transduced cells with an MOI of 12.5 without enhancers, we're seeing around 40% transduction efficiency, and that gradually increases as you increase the vector dose. However, if you compare this to an MOI of 12.5 with enhancers, we see greater than 95% transduction efficiency, which means you're targeting more of the cell population with the vector. When we look at overall VCN in these cultures, again, we saw approximately a 4-fold improvement with our transduction enhancers, apart from when we went to really high levels with the enhancers where it started to become toxic to the cells. But the whole point of this is to try and reduce the amount of vector to the lowest amount possible. The enzyme activity followed a similar pattern to the VCN. So again, saw around 4-fold improvement in a 4-fold improvement in the enzyme activity, meaning we can reduce our vector doses. And we decided to take forward the MOI of 12.5 and 25 with the enhancers to do large-scale transduction studies and demonstrate that we can transduce a large amount of cells with these vector doses. So on the graph on the left, we have our MOI of 12.5 and 25, and we can see an increase in VCN in the pooled CFU cultures. The grey bar is a previous large-scale run we did with MOI of 60 without any enhancers, and we can see our 12.5 with enhancers is equivalent to an MOI of 60 without. Essentially, this means we can use 5 times less vector, which is great news. Transduction efficiency was 100% of both doses. And like the VCN data, we saw very good levels of enzyme activity in these cells. So this takes us on now to some of the results from our first-in-man MPS 3A stem cell gene therapy treatment. So this patient, as a young boy, he was recently diagnosed with MPS 3A. Were approached by his parents at the time, who were very keen on getting him enrolled on our clinical trial, impeding clinical trial, but at that time we weren't quite ready, and he was just a little bit too old for the enrollment criteria. So we decided to treat him as a specials patient under the regulatory authority in the UK. So the details of this patient: he was 2.5 years old at the time of treatment. He has a genotype associated with the most severe phenotype. At the time of treatment, he had age-appropriate development with no significant comorbidities. And he was his cell product was generated at Great Ormond Street Hospital under the specials license. So he was transduced with GMP lentiviral vector at an MO25 within transduction enhancers. He received 13 million cells per kilo, and he tolerated the treatment well. The treatment has been provided by the Royal Manchester Children's Hospital by Professor Rob Wynne and the rest of the paediatric bone marrow transplant team. So the patient tolerated the treatment well and was able to go home 35 days after treatment. And since then, has had no hospital admissions following this treatment, and is doing well so far. So next, I want to show you some of the biochemical data. So we've looked at vector copy number in this patient's cells. So the blue dotted line is the vector copy number of 3.79 of the medicinal products that he received. And we've looked at various different blood cell lineages, the VCN in those different cell types at various different time points over the past 2 years. And we can see that the VCN in these cell types is converging towards that of the medicinal products he received. When we look at enzyme activity, in the white blood cells on the left and in the plasma on the right, the blue dots, the blue solid line is the patient data. The red line is the control, and the blue bar is the normal range, and the dotted green line is the median of that normal range. So that will be common to the next few graphs. And we can see that we're getting an increase in enzyme activity over time, both in the white blood cells and more importantly, the plasma. When we look at heparin sulfate storage in this patient, we can see a rapid decrease in heparin sulfate in the urine and both the plasma. And then when we come to look at enzyme activity in the cerebral spinal fluid, we can see an increase over time. This was slower than in the blood, but it has since elevated above the normal range. And we've seen an accompanied reduction in CSF heparin sulfate. And this is 89% reduction from baseline. So just to sum up the data from this patient, so they've tolerated the therapy very well. We've seen sustained vector copy number over time with supraphysiological levels of enzyme in leukocytes, plasma and CSF, with an accompanied reduction of substrate in the urine, plasma and CSF. So now we're on to our phase 1/2 clinical trial for this stem cell gene therapy. We just enrolled the 5th of 5 patients for this study. So hopefully in the next few conferences, we'll be able to show you some of the data from that particular trial. But so far, it's all looking very promising. I'd just like to touch on some of the other therapies we've developed. So very similar to the 3A, we've adapted the approach for MPS IIIB, and we published this proof of concept study in 2018 in the journal Brain. The study design was very similar to 3A, but this time using a codon-optimized NAGLU gene, which is defective in this condition, with the myeloid-specific promoter. And in this particular study, we're seeing 13% enzyme activity, which is slightly higher than we saw for 3A. And actually, when we've looked in the long term, this actually increases to around 25%. So it seems to be continually going up over time. And we were able to effectively normalize heparin sulfate levels in the brain and correct the behavioral outcome in these mice. We've also developed stem cell gene therapy treatment for MPS II. This was published in EMBO Molecular Medicine in 2018, the proof of concept study. This was slightly different in its design compared to the other 2 approaches I've mentioned. Since this is a disease that affects the skeleton. We've included X-rays in the evaluations 8 months at the 8-month time point to see any correction to the skeleton. With this particular study design, we have a codon-optimized IDS gene, but we wanted to advance the therapy further from previous iterations by linking the codon-optimized gene with the ApoE2 peptide to try and improve transport across the blood-brain barrier. So it's a two-stage approach. Those cells that reach the brain will be able to express the enzyme, and also enzyme being generated in periphery with this peptide should be able to cross the blood-brain barrier to increase levels in the brain. So when we look at enzyme expression in the bone marrow, we see high levels of enzyme expression with both our vectors. It was slightly higher with IDS in the bone marrow compared to the IDS ApoE2, but this is likely down to a lower vector copy number in the bone marrow. Interestingly, even though we have a lower VCN, we see much greater levels of enzyme with our IDS ApoE2 in the plasma. And then we look at brain heparin sulfate. It was only the IDS ApoE2 group that was able to normalize heparin sulfate levels to wild type levels. When we did behavioral studies, we looked at Y-maze and rotarod, and we were able to show coordination and balance are both corrected with IDS and the IDS ApoE2 vectors, but only IDS ApoE2 was able to correct the working memory. When we looked at the skeletal abnormalities, interestingly, all treatment groups, including a wild type transplant, was able to correct the skeletal features. So we measured the width of the zygomatic arch and also the width of the humerus and femurs. So yes, legular stem cell legarival drymal, can't spell, sorry. What I wanted to say that with this data, only ApoE2 is the only one where we've been able to correct both the skeleton and the motor symptoms in the MOS2 preclinical mouse study, whereas IDS alone didn't correct all the conditions. So at the moment, we're in a really exciting position where we've ready to start a phase 1/2 clinical trial, which is due to start at the start of next year, and is funded by AvroBio. So just to summarize, I'd like to say that stem cell gene therapy with lentiviral vectors is very corrective at correcting MPS IIIA and MPS IIIB with no scale-up or distribution assay issues in the brain. Currently, we're well on with a phase 1/2 clinical trial for MPS IIIA, and we've just treated our 5th out of 5 patients. In MPS 2, mice with a blood-brain barrier peptide was required to completely correct the corrective defects with our stem cell gene therapy products using IDS. And based on the preclinical data, it seems that ApoE2 may be required to improve activity of IDS in the plasma and also for cellular uptake. And the phase 1/2 clinical trial is due to start in the first half of next year. And that's it. I'd just finally like to thank, obviously there's a whole host of people who've been involved in these studies. I haven't got time to go through them all, but just a big thank you to everyone who's been involved in Brian Bigger's stem cell neurotherapies group, and all the rest of the teams who've been involved in this work, and all the funding bodies. And that's it. I'd like to thank the audience, and very happy to take any questions you have.
Thank you, Stuart. Thank you, Dr. Ellison, for this wonderful lecture. And we are receiving questions from the audience. Many questions, in fact, mainly about the clinical application, how you see the future of these gene therapies. Dr. Kim Hemsley, for example, is asking about the MPS IIIA patient that was presented. How was the neurocognitive function tracked? How do you suggest to the better way?
Yeah, so we are evaluating the neurological outcomes in this patient. We're currently delayed getting the results, the neurocog results from that particular study due to COVID. What I can say is probably the parents were hoping to for a more effective outcome, I think. But what we can say is neurocognitively, they're doing much better than NPS patients untreated, but not perhaps as well as a patient without this disorder. And I think a lot of these treatments are going to, as we've heard so much so far in this conference, the earlier you can treat, the better. It's from this data, and we can see that particularly in the CSF, it's taken longer time for the enzyme to build up, and is slower for this heparin sulfate to reduce. And if there's already been damage in the brain, it's going to take you're not going to be able to reverse that damage. So interesting, on the clinical trial, we've got some very young patients, one is nearly as young as 6 months old, and some slightly older patients. So it'll be interesting to see if the younger patient does better, has better efficacy down the line than the ones treated later on. So, hopefully, if it's been shown to be successful, preclinical screening is going to become very important. Perfect.
In order to respect the timeline, we've got the 3 other speakers that are waiting. I would like to say thank you, Dr. Ellison, and we will present our next speaker. Thank you for your presentation.
Thank you.
Our next talk will be presented by Dr. Ralph Loffer. Dr. Ralph Loffer obtained his PhD in biochemistry at the Hebrew University of Jerusalem, and he conducted postdoctoral research in molecular neurobiology at the Institut Pasteur. Then he developed a brilliant career in the development of drug discovery in the industry, in the biotech industry, in many companies. Currently, he is the Chief Scientific Officer at Lysogen, and his lecture will be the presentation of the gene therapy study in children with MPS IIIA. Professor Loffer, please go ahead. It's your time. Thank you.
Thank you very much, Hernan, and thank you very much for inviting me to this great meeting. So I'm going to give you an update on our gene therapy study with LIS-SUB-302 in children with MPS IIIA. This is our disclaimer. And just a brief introduction to Lysogen. Lysogen is a gene therapy company active in the field of neurological diseases. And our two lead programs are in lysosomal storage diseases, LysSAF302 for MPS IIIA. That's the program that I'm going to present today. We also have a program in GM1 gangliosidosis that just started a clinical trial, and several discovery projects, including one on neuronopathic Gaucher disease and Parkinson's disease with GBA mutations, a program on Fragile X syndrome, and a technology development program on AAV, novel AAV vectors. So a brief introduction to MPS IIIA, even though most of you probably know the disease very well. The disease is caused to a defect in a gene called SGSH that codes for an enzyme of the same name, sulfoglucosamine sulfohydrolase, or SGSH. And the role of this enzyme is to metabolize heparin sulfate by removing N-linked sulfate groups. And deficiency of this enzyme leads to accumulation of heparin sulfate obviously in every cell in the body, but the cells that are most sensitive to the accumulation, to the toxic accumulation of heparin sulfate, are neurons. And that's why the disease manifests mainly in the brain. And leads to severe alterations, cellular and tissue alterations, and loss of function as I said, mainly in the brain, leading to severe cognitive decline, severe behavioral disturbances, and a shortened life expectancy. And also some somatic symptoms that are relatively mild compared to the other MPS diseases. So Lysogen is active in the field with and has performed several is performing several clinical studies over the years. We started with a phase 1 study called P1. With a predecessor product that was a variant of LysSAF302 called LysSAF301, and this was tested in a phase 1 trial in 4 patients many years, quite a few years ago, and it was a safety trial. And even though it wasn't powered for efficacy or anything, there were some encouraging signs of let's say beneficial effects in most patients, in 3 out of 4 patients, which prompted Lysogen to go ahead and to improve the vector and take this vector the new vector called SAF302, into further development. And we also performed a natural history study called P3, which was a longitudinal 2-year study, which is going to be used as a control group in our gene therapy trial. And currently, we are running the phase 2/3 clinical trial called AAVANCE with LysSAF302. And this is a pivotal efficacy study with a 2-year efficacy analysis. And I will describe this in a little bit more detail in a minute. We also run in parallel a study called PROVIDE, which is a video outcome study that runs in parallel to the AAVANCE study and measures novel exploratory outcomes. So the AAVANCE trial, what we do is we use intraparenchymal delivery of LysSAF302 in order to bypass the blood-brain barrier. The vector is injected into the cerebral white matter, lying just below the surface of the cortex at 6 sites, 3 on each hemisphere. So it's intracerebral injection. The dose is 7 × 10⁻12 VGs per patient. And the inclusion criteria of this trial were developmental quotient, or DQ, greater or equal to 50%, and an age above 6 months. The primary endpoint is the change in DQ measured by the Bailey scale, third edition, at 24 months after treatment. And the comparator, as I said before, is a natural history cohort that comprises about the same number of patients and with the same inclusion criteria. We also measure, of course, secondary endpoints, including behavior. So I mentioned behavioral deficiencies in these children. They are typically very hyperactive and have sleep disorders. And quality of life, imaging biomarkers, and fluid biomarkers. This slide here shows you the composition, so the individual patients, including the age in which they were enrolled. So 19 children were treated, and the age at dosing was between 10 and 65 months. This slide summarizes the safety profile that we have seen so far in our patients. So unfortunately, there was one death. A 43-month-old girl died 18 months after gene therapy administration. And there was no autopsy performed, so we ignored the precise reason of death. But we consulted with experts, and the opinion of one of the experts, an expert in human pharmacology, was that there is no evidence to implicate the gene therapy procedure as a causative factor in the patient's decline and death. This is a quote from the expertise, and I also would like to mention that even though death at this age is not commonly seen in MPS IIIA, it has been reported. So it can happen. And this was, of course, a very unfortunate event. Other AEs, other than MRI findings, which I'm going to describe on the next slide, and concomitant medications reported in more than 2 patients are consistent with MPS IIIA. So typical, let's say, symptoms of MPS IIIA, like for instance, GI disturbances and respiratory infection. There were no clinically significant findings apart from findings related to MPS IIIA in the physical and neurological exams, and no clinical significant lab findings. We did observe, so on MRI, which is of course performed routinely right after surgery and then at 6 months intervals during the trial, we did observe white matter abnormalities near injection sites that were seen on MRI as T2 hyperintensities. And because of these findings, the trial was put on clinical hold by the FDA in June 2020. And we also proceeded with a proactive suspension of the trial outside of the United States, of recruitment of the trial. Currently, we are collecting additional information to better understand the safety profile of LISA-302 and the time course and potential consequences of these MRI findings. What I would like to mention is important to mention that the clinical hold has no impact on the clinical trial timelines, since the main cohort, which consists of 16 patients above the age of 30 months, has been fully treated and followed up, and is being followed up per protocol. And the ongoing investigation and status, what I can tell you so far is that we have not seen any clinically significant symptoms that could be directly attributed to white matter abnormalities. I would also like to mention that MRI white matter abnormalities in neurological practice can be asymptomatic, and they appear to be asymptomatic in this trial. And a preliminary analysis indicates that the process is self-limiting, with the imaging abnormalities stable or diminishing in size from 12 months post-treatment onwards. And we are collecting more data, of course, as we speak. I would now like to switch to the analysis of MPS IIIA biomarkers in the trial. So the primary accumulation of heparin sulfate is used as a pharmacodynamic biomarker by almost every, of course, all the companies who work in the MPS field. We have heard a lot today about heparin sulfate in the CSF, and the FDA in their draft guidance for industry on MPS III, which was published in February 2020, actually specifically say assessment of changes in heparin sulfate concentration in the CSF could provide evidence of in vivo biological activity of the drug, demonstrate proof of concept, and of course help characterize dose response. We are also looking at the secondary accumulation of GM2 and GM3 ganglioside, which are two storage materials that accumulate in MPS III in the central nervous system in MPS IIIA patients, and also in MPS IIIA animal models. And the accumulation of GM2 and GM3 ganglioside is thought to be caused by an inhibitory effect of heparin sulfate on ganglioside metabolism. And this phenomenon is thought to contribute to neuronal damage. Now, of course, as I said, we are all looking at heparin sulfate in the CSF, but it is actually not straightforward. It's actually not really known where the heparin sulfate that is detected in the CSF comes from. And in theory, it could come, as you can see on the diagram here on this slide, it could come from different sources. It could come from the brain parenchyma, which is sort of what everybody hypothesizes, but there is really no really strong evidence for that all of it comes from the parenchyma. And it could also come from the bloodstream. As for instance, it has been reported that low molecular weight heparin sulfate fragments can cross the blood-brain barrier in an in vitro model, and presumably the blood-CSF barrier. So it's not unthinkable that everybody detects the way to detect heparin sulfate is to degrade it into little pieces and then to quantify the pieces, the disaccharides, by LCMS. So some of these could actually originate from outside of the parenchyma, and it could also originate from the choroid plexus. So the choroid plexus, of course, lines the ventricles and produces the CSF, and it is not unthinkable that some of the heparin sulfate actually comes from the choroid plexus epithelial cells. Now, there are no studies really about the origin of heparin sulfate in the CSF. One study that is highly enlightening, actually, that was published last year, is a study by Vera and co-workers from UCLA and Biomarine that saw a 50% reduction in heparin sulfate in the CSF of patients treated with ERT so MPS I patients, treated by ERT. Even though, as was discussed extensively today, the enzyme administered by ERT is not thought to cross the blood-brain barrier, and actually did not produce any neurological effect in these patients. So the authors of this paper suggested that about 50% of the CSF, so that 50% that was reduced following ERT treatment, actually does not come from the brain parenchyma, but probably comes from extra CNS sources. And I don't know, I mean, they hypothesize that it might come from the serum. I'm not sure it comes from the serum. As I said, it could come from the serum as well as from the choroid plexus or from other, CSF lining cells. So our hypothesis is based on that. Hypothesis that about 50% of the heparin sulfate in the CSF does not come from the brain, but comes from extra parenchymal sources. And having said that, now let's take a look at heparin sulfate in the CSF of patients in our trial. So these are preliminary results. We have analyzed on this slide only about 8 patients at 6 months and 5 patients at 12 months after treatment, but we now have more complete data. And the complete data, we haven't really had time to analyze them formally, but I can tell you that they confirm what I'm going to tell you now. We do see about 30% decrease in the pathological heparin sulfate. So when I say pathological, I mean the part of heparin sulfate that is above the level of normal children, which we took from the literature. So there was about 30% decrease in the pathological HS in the CSF, which according to our hypothesis would correspond to about a bit more than 50% of the heparin sulfate that comes from the brain parenchyma. We do not expect to see an effect on heparin sulfate coming from extra brain sources, since we inject our gene therapy product directly into the brain parenchyma, and it will not go outside. And consistently with this hypothesis, we saw no reduction of heparin sulfate in the serum, and we saw no effect as expected on keratan sulfate or dermatan sulfate, either in the CSF or in the serum. Now, regarding GM2 ganglioside, these are the results. We see an about 40% reduction after 12 months in GM2 ganglioside. We haven't looked beyond 12 months yet, and it will be interesting to see whether this continues to decrease. And the same, very similar data for GM3 ganglioside. So as you see here, GM3 ganglioside decreases by about 37% after 12 months, and the decrease at 12 months is stronger than that at 6 months. And again, we will need to see how that evolves with time. So in conclusion, I'm going to conclude here. 19 MPS IIIA patients were treated with an intraparenchymal injection in this trial. The treatment was associated with self-limiting white matter abnormalities near the injection sites, which seemed to stabilize or improve from 12 months post-treatment onwards. And there were no clinically significant symptoms directly attributable to the white matter changes. And LISA-302 led to a CNS-specific reduction of the disease biomarker heparin sulfate. And this is consistent with drug administration directly into the brain, where the accumulation of heparin sulfate causes the predominantly neurological manifestations of MPS IIIA. LISA-302 also led to a reduction in GM2 and GM3 ganglioside in the CSF, and these two storage materials are thought to contribute to neuronal damage in MPS IIIA, and therefore these findings confirm the biological activity and the therapeutic potential of LISA-302. Finally, I would like to thank a lot of people here. So first of all, patients driving the research, the patients, the families, and the entire San Filippo community, in particular the San Filippo Foundation Switzerland, the Cured San Filippo Foundation, the MPS Society, MPS National, and MPS Europe, Casimir Trials, who are involved in our video study, and of course, all our clinical sites. The clinical sites are Amsterdam, UMC, University of Minnesota, Children's Hospital, Wales, Cornell Medicine, Necker, Enfants Malades, Trousseau, Universal Medical Center Hamburg-Eppendorf, the Chalk Hospital in London, Great Ormond Street, Texas Children's Hospital, and of course, all my colleagues at Lysogen that contributed to this work. And thank you very much for your attention.
Thank you, Dr. Loffer, for your great presentation. Very exciting trial of gene therapy. Well, you have shown very good results regarding the biomarkers, but I was very curious, and as many of the families attending this meeting, I guess, we would like to know a bit more about the clinical preliminary results in terms of sleep, behavior, and quality of life of these patients, if you are able to say something about this.
So we took the decision to communicate, to read out the efficacy outcomes other than biomarkers after the 2 years after initiation, after last patient in, which is actually what the FDA recommended in their draft guidance. This being a neurological disease, a neurodegenerative disease, you would really expect that it might take some time to see effects on cognition and on behavior. The FDA in their guidance actually mentions 2 to 3 years minimum. So our trial is going to read out after 2 years, and we would not like to look to sort of make any kind of speculations based on on preliminary or too early data.
Got it. Okay. Rules are rule. Okay. So, well, I'm receiving several questions about this issue, of course, about cognitive function. There's a question about how do you see the future of this kind of gene therapy for older patients?
For older than the ones in our trial, you mean?
Well, if they will some moderate to greater neurological progression, is there any role of this intraparenchymal gene therapy, for example?
Oh, okay. So as I said, we do see some MRI abnormalities near the injection site. So injection site reactions, which appear to be, so far from what we can judge, appear to be without harmful clinical consequences. But we are, of course, very carefully further examining this. And as you know, as in many therapies, including especially gene therapies, it is basically eventually going to be a matter of risk benefit. We do hope, of course, that the benefit that we are going to see at the end of the trial, when the primary and secondary endpoints are going to be analyzed, is going to be significant. And at that point, one will need to evaluate where balance this with some probably asymptomatic injection site reactions.
Okay, I've got a question from Dr. Brian Bigger from Manchester. Did you look at inflammatory markers in the trial in both CSF or serum?
Yes. Yes, we did. So I didn't talk about this, but we, of course, because also, I wouldn't like to say too much about the ongoing investigation in order not to confuse the results. We are still investigating this, but I mean, one of the hypotheses of that was raised, by experts with whom we discussed, and also, of course, internally within the team, was that there could be an immune reaction or an inflammatory reaction against the vector. And I think we can exclude that. So there is there are no changes of inflammatory cytokines or chemokines in the CSF. And there is no evidence, for instance, for either antibodies or T-cell mediated responses against the transgene product.
Okay, thank you very much, Dr. Loffer. And I think we move forward to our next speaker. Thank you.
Thank you.
Next talk will be from Dr. Juan Ruiz. Dr. Juan Ruiz received his PhD in molecular biology from the University of Navarra in Spain and was a postdoctoral fellow at the University of Connecticut. Then he has developed a career in the biotech industry, leading teams dedicated to developing products, including gene therapy products, and today serves as the VP head of European Medical Affairs at Aveona Therapeutics. Juan will talk about developing gene therapies for Sanfilippo syndrome, studies for MPS IIIA and IIIB. So welcome, Juan, and this is your turn. Thank you.
Thank you so much, Dr. Amartino. And I would like to start thanking the organizers of this important symposium, particularly Jordi Cruz, for giving us the opportunity to present the programs that the Aveona is developing for the treatment of MPS IIIA and IIIB. My name is Juan Ruiz, and I'm the medical monitor responsible for these clinical trials. And in this presentation, I'm going to focus on the most advanced program we have for MPS IIIA. This is called Transfer A. And I'm going to present some, for the first time, some relevant data, for example, brain MRI data. This is my safe harbor statement. And we have reviewed during this presentation and other sessions in this meeting MPS III. These are, in fact, a group of 4 different genetic diseases, very similar because all of them lack an enzyme that is required from the breaking down and degradation and recycling of a complex sugar molecule, heparin sulfate. You see here in this picture a cell from a child with Sanfilippo with these little bubbles that are the lysosomes where heparin sulfate has accumulated. These accumulations leads to cell malfunctioning and ultimately to cell death. MPS IIIA is more common than the MPS IIIB, and both of them, MPS III in general, affects more the central nervous system, where there is a slowing and regression of development in cognitive capabilities, but there are also important other features like impulsivity, hyperactivity, sleep disturbance, behavior abnormalities, including autism-like symptoms. With time, children may develop seizures. And in general, I mean, a common feature is that all children lose continuously skills progressing to dementia. Although not as frequent as in other MPS diseases, physical features are also important because they might help early diagnosis of the disease. The most prominent might be the coarse facial features with excessive hair growth or frequent middle ear infections that also might affect the development of children. There is unfortunately no treatment for this devastating disease, and children die usually during the second decade of life. Aveona has developed 2 AAV9-based vectors, AAVs are viruses that infect human and primates, not causing any known disease, and they can be modified to replace the viral genes by therapeutic genes that inserted or included in these viruses and upon injection in the subject. And in this case, with AAV9, upon intravenous administration, they might reach the brain, express the enzyme within neurons and other cells within the central nervous system, correcting those cells that have been transduced. But they can also release the enzyme, and this enzyme may be uptaken by neuron cells with this cross correction that has been mentioned in other sessions. Aveona has 3 clinical programs for MPS IIIA and IIIB. 2 programs for MPS IIIA, called Transfer A and ABT003. In Transfer A, the targeting population are children at early stages of the disease, either from birth to 2 years of age, or those that are older than 2 years with a developmental quotient equal to or higher than 60. ABT003 on the other side is targeting children with middle and advanced phases of the disease, not candidates for Transfer A. And so in general, those who have a developmental quotient lower than 60. Both trials are being conducted in the US, in Spain, and in Australia. On the other side, for MPS IIIB, Transfer B study targets a similar population. It's very, very similar in many aspects to Transfer A, and is targeting children at early stages of the disease. The trial is being conducted in the US and in Europe in the sites in France, Germany, and Spain. Both Transfer A and Transfer B trial share many features in the clinical trial design. They are single dose, open label, meaning no placebo, dose escalation trials. They are both recruiting now in cohort 3, the highest dose. And the product is administered intravenous during 15 to 45 minutes. Children usually stay at the hospital for 2 days. And from day -1, steroids are used. And then for the next 3 months, including 1 month for heparin on the steroids, are used in order to prevent immune responses that might have safety, that might induce safety adverse reaction. The duration of the study is 2 years, but all children finishing 2 years in Transfer A and Transfer B are enrolled in a long-term follow-up study for additional 3 years. So a child enrolled in Transfer A or Transfer B is going to be followed for efficacy and safety for 5 years. The primary endpoint is the cognitive scores that are obtained in children treated with the product compared with untreated children in natural history studies. Obviously, these are phase 1, phase 2 studies of product safety is of paramount importance in the evaluation. And then there are a number of secondary endpoints that are being used in order to address behavior changes or potential changes with the treatment, quality of life. We measure enzyme activity, biomarkers, systemically and in the CNS, and also we conduct brain and liver MRIs to see changes in those relevant organs. Key participation details of these trials. Most visits, except those around treatment, are outpatient visits. And all participants, as I mentioned before, receive a single intravenous administration. We are enrolling children from all countries around the world and providing, obviously, support for the families, up to 4 family members with lodging, meals, transportation, but also translation in case that the families don't feel comfortable with the site that they are directed. And as I mentioned, we have sites all around the world. During those visits that are initially more frequent, and then every 6 months, in the main visits, we conduct lumbar puncture to collect CSF. We do perform MRIs for abdominal and brain MRIs, and then cognitive scales that are relevant for the primary and secondary endpoints. The patients are followed by experts on the disease, and during COVID-19, we've been following all international, national, and site-specific instructions in order to ensure patient and family safety during the trial, but also focusing on the integrity of the data. This is an update on enrollment of both trials, Transfer A and Transfer B. 33 patients have been screened, 21 of them have been dosed, and 15 patients have been dosed in cohort 3 with a higher dose. 10 of them have been enrolled under the enrollment criteria involving less than 2 years of age or a developmental quotient equal or higher than 60. Children from cohort 1 and 2 already are being followed already in the long-term follow-up, as well as some children in cohort 3, but the median follow-up in cohort 3 is 32.6 months. Regarding Transfer B, 19 patients have been screened, 11 of them have been treated and dosed, and 4 of them in cohort 3. And these are the median follow-ups for different cohorts. With this introduction, I would like to review some of the key data we have collected so far in Transfer A, as I mentioned at the beginning. This is a very important slide because here we are presenting data from children, cognitive age equivalent data from children enrolled in cohort 3 with a higher dose, and comparing this data with the data that we have observed, we have collected in natural history studies. So this, I hope you can see here in light gray, the data from 99 children enrolled in 4 different natural history studies that represent our comparator group. In blue, we have the data from children in cohort 3. In the horizontal axis, we represent children age, and the vertical axis represent cognitive age equivalents in months. An age equivalent is the functioning age of a child, and is the parameter that we are assessing using neurocognitive scales in the trial, and this is the primary endpoint. The DQ 100 line, the developmental quotient 100 line, represent the expected normal development, and is the ratio between the age equivalent and the children chronological age. This black solid line here shows the average typical progression of children with MPS IIIA, as estimated from all the data collected in natural history studies. The children with MPS at the beginning, in the early times, they develop close to normal, I mean close to the DQ 100 line, and 1 year and a half, 2 years, they start deviating from this, and they reach a plateau in the acquisition of cognitive skills between 30 to 36 months, and after 4 years, 48 months of chronological age, they continuously lose skills over time. Compared with this, we are reviewing here the data collected in cohort 3, and we can very clearly see two different populations separated by the DQ60 line. Those on the right of the DQ60 line were treated when they were older, and with the exception of this child that shows a stable evolution, they are evolving similar or no different from natural history represented by the black solid line here. But in this slide, I would like to focus on the population of the children that have been enrolled at younger age with DQs higher than 60. And they evolve, as you can see in the slide, different from what would be expected according to natural history. So one child here that was treated at 27 months of age and has been followed for 3 years after treatment has achieved a cognitive age of 49 months at the age of 64 months of chronological age, something that has not been described in natural history for any child with a severe phenotype. Another child that was treated at 18 months of age and followed for 30 months, 2 years and a half, is also gaining skills and separated and deviating from natural history. This child has 2 known mutations, is an extremely severe phenotype, and still is progressing, deviating from natural history. Similarly, a child that was treated at 1 year of age, that has been followed also for 2 years and a half, 30 months, has achieved a cognitive age of 44 months at 42 months of cognitive age, tracking along the DQ 100 line and clearly deviating from the expected evolution described in natural history. There are other children that have been dosed up to, as I mentioned, 10 patients with these clinical characteristics, and we need additional follow-up in order to confirm these highly encouraging results that I have described from these 3 children. With the permission of her parents, I would like to show you a video recorded as part of the cognitive evaluation of the child that was treated at 1 year of age. The evaluation was conducted at the last visit, 30 months after treatment. And let me play the video, hopefully, okay. I don't know if you hear, okay, unfortunately, I don't hear sound, and I would like to check with the organizers, I mean, with the IT personnel, if they can make sure that you hear. Oops. This is unfortunate. Can you please, someone from the organizers, check that the sound, maybe something that I have done that was not, okay, yes. Okay, thank you. Yeah. Okay, I'm really sorry because in this video, and maybe for the sake of time, I don't know if we will have time. I would love to have the opportunity. Okay, thank you. Okay, it seems that the organizers have been able to solve the problem with the sound. Okay, so let me try to play the video and see if it works.
Off the last little bit of this thing called the mullen, and then we'll do some games. Okay.
Do you want something else to eat? Oh right.
Just been eating morning tea.
Literally just ate until you said I was finished.
So, what do we do when we're hungry?
I got my lunch.
Yeah. What do we do when we're hungry? Okay, I'm going to leave you focus on that because I think you're going to...
Oh, that was supposed to be for later. Mummy forgot I put that in there as a treat for when we left.
Mom, go for me.
I did get it for
We'll
you.
leave that one later though.
Later.
Yeah, that's for when we're finished.
That's for when we're finished.
Yeah, after dinner.
After dinner.
Is Tilly likely to want to eat all of that and focus on that for an hour? Because that could take about 20 minutes, 30 minutes.
No, that will take about 2 seconds.
She doesn't eat the crust.
She doesn't eat the crust.
She eats the inside of the...
I'm going to food.
Okay, cool. I wanted to do some...
No, not yet.
There's something left in there.
Because I thought, Tilly, if we're just eating, we might have other stuff to do.
I have chocolate.
No, that chocolate is for later.
That's after dinner. Well, you already had the Freddo Frog.
No.
You have too much chocolate?
I want to have more chocolate.
Do you ever have enough chocolate?
She does actually stop, but she's finished. Can you have half of that chocolate?
Huh?
Do you want the rest of your sandwich?
I want half of it.
You can have half of it.
Okay.
Can you break it in half?
Are you going to share some of that?
Okay, so sorry for the interruption. I thought it was interesting to see the video because what seems normal in this slide is pretty remarkable. And you can appreciate the spontaneity, the gestures, the understanding of the questions and comments, and the quick reactions and the complicity with mom and dad, as well as the language and motor coordination. Remember, this is a child that was treated at 1 year of age, and this is 2.5 years after treatment.
Finish off the last little bit.
Oops. Changing now to biomarkers, we are measuring as secondary endpoints of transfer A biomarkers, both systemically and also in the CNS. And this slide summarizes results that show a consistent, rapid, and sustained decrease in biomarkers in the CNS. We have here CSF heparin sulfate, GM2, GM3, but also systemic biomarkers like plasma heparin sulfate or urinary GAGs. Normalization of GM2 and GM3 that is statistically significant is particularly relevant because they are secondary substrate that has been described in previous talks that are involved in the pathology of the disease. Finally, I would like to show you some brain MRI results from the 3 young children that I have highlighted during the review of the cognitive results. These results correspond to 2 years of brain MRI images, and we are presenting here a result of gray matter, corpus callosum, and amygdala. Children with MPS III show characteristic brain MRI changes that can be exemplified here in this picture. Is small, but hopefully you will be able to see the characteristic features of brain MRI in children with Sanfilippo. This image shows brain atrophy with a decrease in the gray matter, with widening of cortical sulci, separation between these, as well as decrease and thinning of the corpus callosum, and an increase in the ventricles and subarachnoidal space, that is an ex vacuo increase due to the brain atrophy. All these changes indicate brain atrophy. In addition, other changes described in brain MRIs from MPS IIIA include decrease in the amygdala volume. Changes in gray matter or amygdala volumes have been correlated with relevant features and symptoms of the disease, such as the decrease in DQ or lack of fear, respectively. So looking at this graph, and this graph is taken from the natural history study conducted by Dr. Shapiro, and this corresponds to gray matter volume. In black, you see the decrease in the gray matter volume in different patients across different ages. And we have plotted here the results for the 3 children that I have described previously. You have one here in red, one in blue, and one in green. And these children shows an initial decrease that is suspected because of the use of steroids during 2-3 months after administration, that later on recovers and continually increases in volume compared with the decrease observed in gray matter in natural history. Corpus callosum here, represented here, and amygdala represented here, are represented, and each line represents a single patient followed for 2 years. As you can see here, there is a continuous increase in corpus callosum volume, in amygdala volume, something that is compared with what has been described in natural history. Finally, I would like to review with you this brain MRI image corresponding to the child that was treated at 1 year with 2.5 years follow-up, and this image was taken at 42 months of chronological age, to compare with this image also obtained at 38 months, very similar age, without the treatment. And what the radiologists informed from this brain MRI is that there were no abnormalities in the white matter, gray matter, ventricles, amygdala, corpus callosum, and other areas of the brain. So what this brain MRI indicates is preservation of brain parenchyma and normal brain growth 2.5 years after treatment. With this, I would like to summarize the results that we have obtained so far in Transfer A. I didn't mention that is included here that the safety of ABO-102 so far the product has been very well tolerated, and there are no deaths or no drug-related severe adverse events. And those drug-related adverse events have been in general grade 1 or 2 and are all resolved. They involve mainly changes in liver enzymes, ALT and AST, without changes in bilirubin or other markers that might indicate liver dysfunction. There have been some changes in platelets in 4 children, as has been described with other programs, although to a much lesser extent, and the lower levels has been 69,000. And there have been no relevant immune responses against the capsid, as indicated by ELISpot. As I have presented, children treated before 2 years of age or a DQ higher than 60 track along normal development and show continuous developmental progress 2.5 years, 3 years after administration, when they would otherwise be experiencing cognitive declines according to natural history. I have presented data from this child that was treated at 1 year, that is 2.5 years later, showing consistent normal development with brain MRI that is showing basically a normal brain MRI. And I have also summarized the results of biomarkers on the CSF level for heparin sulfate, GM2, GM3, and reductions also on systemic heparin sulfate and urinary gas. With that, I would like just to thank all the patients and the families that have participated in the trial, the MPS community and foundations, and all the people in the different sites across the world who participate in our trials and have made this possible. These are just finally the contact details, and I'm ready to answer any questions.
Thank you, Juan. Congratulations for your nice talk. I'm receiving here some questions from the audience. My friend, Professor Michael Beck from Germany, well, he is asking the same question I had prepared for you. that is why and how does your construct overcome the blood-brain barrier? Because it's clear that it passes the barrier, but what's the mechanism? Is there any window maybe that lapse of time that should be considered? What about this point? Thank you.
Okay, thank you for the question. And well, this is something that not only us, but other groups, both in preclinical, in animal models, different animal models have shown AAV9 is able to cross the blood-brain barrier. It is true that most of the vector that is administered systemically goes to the liver and other organs, as Dr. Wilson has presented previously in a session, but there is a small percentage of the virus who cross the barrier. And working with the doses that we are administered, this dose is able to distribute what we believe evenly in different regions of on the brain, express the enzyme and reduce biomarkers. And as we have seen, and we believe this is related with the treatment, improve, facilitate cognitive gains and development and prevent and preserve the brain architecture as we have shown here. There are other groups, and a lot of the studies indicated that the proportion of the vector is small, but with the right doses is sufficient to reach the brain and distribute and express the enzyme.
Okay. I've got a question from Dr. Kim Hemsley. He says, given the outcomes in MPS IIIA patients treated under 30 months of age, what are your views on the implementation of newborn screening for this disorder? Good point too.
Yeah, absolutely. This is a very important point, and has also been discussed during this symposium by some of the experts, and in fact the question has been presented to other groups. It's newborn screening will be paramount in order to diagnose children at a time when the treatment might be most efficacious. We know that there are these ethical issues regarding diagnosis early diseases who have no treatment, and what might be the consequences for the parents, and even the potential misdiagnosis or mild phenotypes that might lead to a much less severe disease and have the parents being worried about this. But overall, I think that the possibility to diagnose early, to intervene early, as we have seen here, not only this, but other neurodegenerative diseases, overcome clearly those hurdles and will allow the possibility to prevent, but what is more important, allow normal development of children.
Okay. Now, I've got also a comment or question from Janet Ojeda from Red San Filippo Foundation. Janet is asking about the extension, the advance of the extension research in other subgroups of San Filippo patients, including those with antibody to the enzyme and antibody to the vector, to the AAV9. Could you please say something to the foundation?
Yes, no, absolutely. I haven't presented here results from Transfer B. There is a poster in this meeting where we summarize the results. The Transfer B is the trial for MPS IIIB is less advanced. It started later and has enrolled less number of children. We have the data showing similar changes in biomarkers. It's still early to see the cognitive evolution on that trial. So we will update this in due time when more information is available. Regarding the possibility to treat children with antibodies to vectors, this is one of the problems, and this is one exclusion criteria in our trial. There are several groups, including companies, developing potential interventions that might decrease the level of antibodies, even eliminate completely the antibodies in circulation for a window of time that might allow the intervention and the administration of vectors. Those are very interesting studies that have been presented, that have been applied in other diseases like kidney transplantation, that might be applied in the future in gene therapy. There are other ways to also immunomodulate and prevent the development of antibodies and allow second administration. So this is an area of investigation that is really important for the field, not just for this program, but for all programs involving systemic administration that might allow the possibility that children with antibodies access to these therapies, and also to reduce potential safety issues that have been associated with the early development of antibodies to the vectors.
Well, thank you, Dr. Ruiz, for this wonderful lecture and discussion, and we will move forward to our next speaker.
Well, bye.
And our next speaker is someone local, because as you know, we are supposed to be in Barcelona right now. Maybe you can feel it, but the Rambla is out there. And our next speaker is from here. Is Merce Pineda. Merce is an outstanding pediatric neurologist from Sant Flor de Déu Hospital in Barcelona. And she's also a clinical researcher with extend an impressive career developing strategies to better care our patients with neurometabolic diseases. She is especially involved in the neurodegenerative lysosomal diseases in the last years, and she's part of the organizing committee of this meeting. So I would like to thank Merce for all these efforts. She will talk about safety and tolerability after intracerebral ventricular administration of adeno-associated viral vector serotype 9 containing the human sulmidase gene, ongoing phase 1/2 clinical trial in Sanfilippo syndrome type A. Merce, welcome, your turn now.
Thank you, Hernan. I shall try to do my best. Oh, let me see. So, no, the slide don't pass. Oh, yes. Well, all of us at this moment know what is Sanfilippo. Of course, if we have a mutation in the gene, we have the deficiency in the enzyme, and of course it affects especially the brain, but also it makes a neurodegeneration with cognitive decline, severe behavior disturbance, and unfortunately a short life expectancy. Oh, wait. But gosh, no, not yet. But it also affects the, no, this. It also affects peripheral organ tissues. These are less affected, but in some of them, facial features are very predominant. Some of them also have recurrent diarrheas, recurrent ear, nose, and throat infections. In the visceromegaly, hepatomegaly is also quite important in some of them, but it goes up and down. But what is important in our trial is the route of administration. So this was suited for monogenic genetic disease and optimized the delivery to the main target organ, that was the brain. The intracerebral ventricular administration into the lateral cerebral ventricle, the dynamic of the cerebrospinal fluid circulation allows exposure of the adenovirus in time to the complete brain ventricular system and subarachnoid space, and envelopes all the cortical convexities of the brain. The cerebrospinal fluid is ultimately absorbed in the vascular system, and an amount of the intracerebral ventricular administered product also passes into the bloodstream, reaching the liver, that can also produce and secrete sulfamidase enzyme, which reaches all the peripheral target organs. See? So the intracerebral ventricular administration was suitable. It was a standardized surgical procedure using standard techniques, materials, and equipment with well-known and manageable risk that you have seen that showed Dr. Candela. It provided a widespread distribution in the whole central nervous system, primary target organ of the disease, reaching also the periphery. Due to the central nervous system immune privilege, there is a reduced likelihood of serious immune response, and there is no need for concomitant immunosuppressants. Validated in preclinical studies in mice and dogs, the intracerebral ventricular administration corrected both central nervous system and somatic pathology in mice. So we started the trial. We have to say that it is a unicentric, non-randomized, open-label, parallel group, phase 1 and 2 of the study. The main objective of the study was to look to the safety and tolerability after the intracerebral ventricular administration with a single dose vectors and 3 doses cohort of patients with MPC3A. It was performed in a hospital, Hospital Sant Joan de Déu of Barcelona, and I was the principal investigator. It doesn't move. I have to say that in the screening, we found quite a lot of patients that had antibodies to the adenovirus 9. So we had to do a lot of work. So the recruitment period finalized in November 2020, and 9 patients have been treated now in the follow-up period. You can see here the 3 doses in the 3 cohorts, and the 3rd dose is double the 2nd dose. Male and female MPS IIIA children were between 2 and 5 years of age. One of the criteria inclusion was that they had to be more than 2 years of age, so to express the disease. And what is important also, it was that they had to have 1 missense mutation. All the children were treated according to the protocol under general anesthesia, American Society of Anesthesiologists, physical status classification, and all children patients with severe systemic disease. The neurosurgical intervention was quite short. The duration was 30 minutes to 1 hour, and no incidents were reported during the neurosurgical intervention during recovery. Just before the neurosurgical intervention, we put a portacat to all the children because during the first year of treatment, we have to take blood samples quite a lot of times. So this is an easy way, and it's not harmful for the patients when we have to take the samples. Another thing is, according to the protocol, the patients were transferred to the intensive care unit. The length of stay in the intensive care unit was from 15 hours to 22 hours. And one of the parents was with the patient in the intensive care unit room. No incidents were reported during the intensive care stay, apart of a vomit with the first tolerance of water, secondary to anesthesia in 1 patient. Families are very grateful when they can be with their children in the intensive care unit. And afterwards, they were transferred for 24 more hours to a room. Of course, due to the pandemic COVID, we had to be very careful. And general safety, the intracerebral ventricular administration is uneventful. They immediate postoperative period has a prompt discharge. The clinical status is stable. The standard clinical hematology, biochemistry, and urine analysis didn't show any relevant changes, and the electrocardiogram and vital signs didn't also show any relevant changes. So what shows our specific safety is that the neural immune response was as expected, rising the total and neutralizing antibodies against adenovirus 9 in serum and cerebrospinal fluid, but not against the enzyme. The cellular immune response, when it appeared, it was transient, of very low intensity, and without any clinical translation, so we didn't require any additional intervention. The adverse events that we only had one serious adverse event related to the intercerebroventricular procedure. It was a cerebrospinal fistula secondary to subcutaneous suture stitch intolerance in 1 out of 9 patients. It wasn't related to the vector at all. This patient was asymptomatic during the whole episode and received only surgical repair. And this also, sometimes it can happen when we put a valve for hydrocephalus. So it's not a strange thing that could happen. No other serious adverse events have been reported. The only reported treatment emergent adverse events were non-serious, mild intensity, transient, and assessed by the investigator and sponsors, and not related to the adenovirus virus treatment factors, but to baseline disease or to pediatric concurrent disease. We know that they have a lot of nose infections and throat infections, these children. The safety conclusions that we have is that the treatment with adenovirus has displayed an important safety and tolerability profile. Altogether, this profile and safety data collected show no identifiable safety trend or signal and were consistent with baseline disease-related events or other concurrent pediatric disease. Cerebroventricular administration targeting the central nervous system, the main affected organ in MPS IIIA patient, presents a positive benefit-risk profile. And finally, what I have to say is that due to the COVID pandemic that we have had during this study, all the biochemical and clinical results we couldn't be able to do because the children couldn't come to Barcelona, but we expect that now we shall be able to finish with all the controls. And in the next symposium or next meeting with MPS or lysosomal disease, we shall be able to show to you. Thank you very much for your attention. And these are our places in Barcelona. Thank you.
Thank you, Merce. Very nice talk about this gene therapy trial. We are a bit late, but I'm waiting for some questions from the audience. I would like to congratulate your work, not only this particular subject you have presented, but all the organization of the meeting. We're supposed to be finishing 10 minutes ago, and we have to move forward because there's a symposium in 20 minutes. And so if there's no questions for Merce, I would like to thank all the speakers, this marvelous, wonderful session about new therapies. We have to say that neurodegeneration in MPS is the big Cinderella. It's still a big challenge and meet need for many patients around the world. And we can say that many advances have been done. This session is a very good example, a clear example of that. There are more than few therapies being developing, preclinical and clinical trials ongoing. And we hope that next MPS Society meeting in 2 years, we will show more results, and there's a good hope for all the community, MPS community. Thank you to everybody, and I invite you to stay online for our satellite symposium. Thank you everybody for your attention, and enjoy Barcelona.
Thank you.